So the next speaker is John Turchi from Indiana University School of Medicine. Long time no see, John.
Well, thank you very much for the introduction, Jim. Good to see you again. And Karen, very nice talk. We'll talk after this as well. I have some questions, and I'm really excited to tell you about some of the work going on in the lab. It was 2 years ago I gave a talk at this conference on our RPA inhibitors, and today I'm going to tell you about another target that we've been looking at, which is COO, the protein involved in non-homologous end joining. And I'll tell you about some of the small molecules inhibitors we've developed against COO as a novel mechanism for DNA PK inhibition. And so as we're all aware, the DNA damage response relies on a series of protein DNA interactions. There's been a lot of effort and a lot of enthusiasm around the PI3 kinase like kinases, ATM, ATR, DNA PK. But importantly, all of these require a protein DNA interaction to serve as an essential step to activate these kinases. So in the case of ATR, you won't get activation of the kinase and signaling to the downstream events occurring unless RPA is bound to single-strand DNA and then forms a complex with ATRP and other proteins to allow ATR activation. Similarly, ATM cannot be activated unless mRN is bound to that DNA duplex, the strand breaks or this DNA discontinuities to allow ATM activation. And also, obviously, activating the DNA PK catalytic subunit requires the central COO7080 heterodimer to bind and allow activation of the kinase. And so considering the enthusiasm for targeting these kinases in the context of cancer therapy to take advantage of potential synthetic lethal interactions, often seen in a number of cancers that have alterations in the DNA repair or DNA damage response proteins, there's been a fair amount of effort towards targeting these PI3 kinase-like kinases. But we would argue that if you're looking for developing a specific inhibitor, while the kinase active sites all have a great degree of homology, and trying to find a small molecule to fit into that ATP binding pocket that would allow specificity between the three is a challenging undertaking. And we would argue if you look, in fact, at the protein DNA interactions, you have the potential for considerably greater specificity. So the RPA single-strand DNA interaction formed by the OB folds of RPA allowing the single-strand DNA to fit in that is required for ATR activation looks completely different than, say, the MRE 11 RAD50 complex with a duplex DNA, which looks very different than a COO70, COO80 interaction with its duplex DNA required for DNA PK activation. So we thought the potential of targeting these kinases through their DNA binding components had some utility. So if we're looking at COO protein, the one we'll talk about today, it has this very unique structure shown here by Jonathan Goldberg and complex with the DNA duplex. And we'll talk a little bit about trying to target that molecule as a mechanism to impact the DNA double-strand break repair pathways as well as the DNA damage response and really help us elucidate some of these pathways. So you can imagine, obviously, if we can create a small molecule that fits within the active site of COO to block it from binding DNA, we would necessarily block non-homologous end joining, the step of allowing that first initiation event to then catalyze the recruitment of DNA PKCS, and then ultimately strand ligation to form a duplex from that double strand break. I just told you about the DNA damage response. We'd obviously expect the effect on non-homologous end joining, but also the potential for activating ATM and crosstalk between those two pathways and potentially pathway choice. In addition, a small molecule inhibitor of the COO DNA interaction could have pretty unique effects on telomeres. COO is known to interact with telomeres and protect the ends and potentially block nuclease activity in events that are independent of the catalytic subunit of DNA PK. So a small molecule COO inhibitor could have some unique aspects and allow us to elucidate its roles in telomere metabolism as well. So we set out to try to block this COO DNA interaction with small molecules. We term these molecules COO DBIs, or COO DNA binding inhibitors. And I'll tell you about a group that we found focused on this X80 molecule we initially identified, in fact, as an XPA inhibitor, looking at that protein DNA interaction. as with any good medicinal chemist, you want to demonstrate some degree of specificity. So we tested this molecule against every protein we had in the freezer. And at that time, there were about 20 DNA binding proteins. And this molecule showed great specificity for XPA, except for one alteration, and that was Ku. We found that it could, in fact, inhibit the Ku DNA interaction pretty well. So what we did was dock this into the Goldberg crystal structure and really identified this pocket where this benzoic acid was sitting that has a potential for further optimization. So we made a series of molecules that contain this methoxybenzene ring that fits very nicely within this pocket to create the 68 compound, which just has this methoxybenzene. We were then able to reduce this double bond here to a single bond to create the 149 compound. Doing a little more medicinal chemistry to make this something that is a little more amenable to cellular activity and potential in vivo analysis. We changed the methyl group to a trifluoromethyl to create the 322 compound. And then finally, I got rid of this carboxylic acid completely and replaced that with a tetrazole, which has similar electronic configurations, allows similar hydrogen bonding without the charge, and gives you essentially a carboxylic acid mimetic, or a bioisoster, as we call it. So we could take all of these molecules and demonstrate pretty effectively that they could block the Ku DNA interaction. So in a standard electrophoretic mobility shift assay, purified DNA in a test tube, we add purified Ku heterodimer, purified from a baculovirus expression system. You can get a change in mobility of that DNA, and that's the bound product. Titrate in the small molecule inhibitor, and we can decrease that binding pretty effectively. When you plot the percent binding activity versus the compound, you get nice titration curves and can calculate IC50s. So just simply putting on that methoxybenzene off of this carboxylic acid, we were able to increase the potency and get an IC50 down into the 9, 10 micromolar range compared to 75. So a pretty significant increase in potency. And that increased when we reduced that double bond to a single bond here in the 132. And both the 322 and 245 were even more active as Ku inhibitors. So for inhibiting DNA binding activity, you might imagine we could inhibit kinase activity. So we ran a standard DNA PK catalytic assay using a synthetic peptide substrate, all dependent on DNA in the reaction. So now instead of just purified Ku, we have the purified heterotrimer, and we can get very potent inhibition of phosphorylation activity, the kinase activity of DNA PK across the board. Here shows the X80 parental molecule. We get a much greater potency for both of these two compounds, the 68 and 149. And then those further modifications take it even further, just looking at, we've changed the x-axis here. So now we're in the few hundred nanomolar IC50s for these molecules. So we were able to effectively inhibit the kinase through inhibiting the protein DNA interaction of Ku. For both of these experiments, we use the NU7441 control. This is a commercially available DNA PK inhibitor that targets the catalytic subunit, the DNA PK CS 460 kilodalton subunit. And so it is a very potent inhibitor, about 30 nanomolar in these assays. When we put that in the electrophoretic mobility shift assay, it had absolutely no activity there, as expected. So that kind of serves as our negative control and really shows that the molecules we've discovered are acting to inhibit DNA PK through a novel mechanism, which is blocking that Ku DNA interaction. And so as long as we had that, the potential is that these molecules could either bind the Ku protein or bind DNA. The last thing you want is another DNA binding molecule in our arsenal for chemotherapeutic agents. And so we set out to ask that question using a thermal stability shift assay, which takes advantage of being able to measure a protein's denaturation effects over temperature, essentially allowing you to determine a melting temperature at which half the protein is denatured. The model being when you have that protein then mixed with a small molecule or drug that binds to that protein in some context, you create additional hydrogen bonds, ionic interactions, hydrophobic interactions, van der Waals interactions that stabilize that protein and essentially increase the melting temperature. So you get a thermal shift, or a shift in the thermal stability of that protein. So we did this again, purified protein in a test tube with the Cu7080 heterodimer, measured the melting temperature in just vehicle control, and then in the presence of the 245 inhibitor, a fixed concentration of 10 micromolar, and end up showing a pretty significant 6-degree shift in the melting temperature. So this told us that, in fact, the small molecule was, in fact, binding directly to the protein to elicit its activity. We've also run and published previously this entire series of compounds don't bind to single-strand or double-strand DNA using a fluorescence displacement assay. So it really served as a good convincing evidence that the molecules we have developed, in fact, are binding to the protein, not DNA, to elicit the activity. So we've been able to measure inhibition of Cu binding in a test tube, inhibition of phosphorylation activity of a synthetic substrate. But in fact, the best known substrate for DNA PK is in fact the catalytic subunit itself, the autophosphorylation of DNA PK CS, an essential process in non-homologous end joining. And these autophosphorylation events occur in 2 flavors, one at the PQR cluster, which is a series of 6 serines located between amino acids 2023 and 2056. And importantly, there's a very effective commercial antibody available to measure that phosphoserine residue. The other autophosphorylation events are in this ABCDE cluster. That are also important for conformational changes and complex disassembly. Both take place during the normal non-homologous end joining and also are involved in potentially regulating pathway choice between homologous recombination and non-homologous end joining. So in this assay, in a negative control, in the absence of DNA on this Western blot, there's essentially no autophosphorylation. You add DNA to the reaction, you can get potent autophosphorylation of the catalytic subunit. When we incubate with our inhibitors, again, pre-incubation of the inhibitors with the protein prior to the addition of DNA, we can get very potent inhibition of the autophosphorylation activity, something you see with the catalytic subunit as well, but not with 329, which is a completely different structure, chemical structure that inhibits other DNA protein interactions, specifically the RPA DNA interaction, but it has no effect on the autophosphorylation because it's not a Cu inhibitor. So having demonstrated that, the real question is, can we inhibit non-homologous end joining? So in collaboration with Les Hanakahi at the University of Illinois, she ran a series of assays for us using the in vitro analysis measuring multimerization of a linearized plasmid DNA that she's radio-labeled, shown here. When you put a cell-free extract containing every protein in the cell that she could get out that's competent to catalyze the non-homologous end joining, you measure these multimers. So she ran duplicates of the control and then increasing concentrations of the 149 inhibitor in this case, and was able to measure efficient inhibition of non-homologous end joining. Although the potency wasn't nearly as good as what we were seeing in the assays performed with just the heterotrimer, or in fact purified Cu, we were getting reasonable inhibition over this concentration range. And we attribute that change to really a non-specific protein interaction that we can talk about a little later. What this showed us, though, is that we are able to inhibit Cu in a much more complex reaction mixture, a whole cell extract or a cell-free extract, compared to just purified protein in a test tube. The next question was, could we really do this in cells? And so for this assay, we invoked a host cell reactivation assay, essentially a linearized GFP plasmid where you separate the CMV promoter from the GFP structural gene, transfect that along with a control circularized plasmid into cells that expresses DS-red. Only after a recircularization event where that linear DNA is recircularized will you be able to get GFP expression. So then you can measure both green and red through flow cytometry and calculate the NHEJ efficiency. In our case, we normalized everything to 1, but in our cases, this was about a 30% efficiency of non-homologous end joining compared to the DS-red. But what we show is that pre-incubation of the cells with either any of the compounds over the entire group were in fact pretty potent in inhibiting non-homologous end joining in cells. So we're able to now treat cells with a small molecule, have it get into the cells, bind to Cu, and inhibit Cu's activity in non-homologous end joining, ultimately giving us this decrease in NHEJ catalyzed rejoining events. So we were pretty excited about that. We could get this thing into cells. One of the questions was, are we really going through the target we had? And if in fact we're inhibiting non-homologous end joining of this synthetic transfected DNA, presumably we would be able to block non-homologous end joining of double-strand breaks induced by other agents. So we performed this reaction, this experiment, looking at bleomycin sensitivity in a series of cell lines, bleomycin being a radiomimetic that can induce DNA double-strand breaks. And in a wild type MEF cell line just treated with vehicle, you get a pretty standard titration curve, an IC50 somewhere around 12 or 14 micromolar on its own. When we do a concurrent incubation with, in this case, the 68 compound, we can show an increased sensitivity to that bleomycin. So we are inhibiting non-homologous end joining, catalyzed repair of these bleomycin-induced double-strand breaks, leading to cell death. So we get about a 4 to 6-fold increase in potency of the bleomycin, or a sensitization effect. The question is this really a Ku-dependent effect, or is there something else going on by treating these cells? So if we take a Ku80 null cell just treated with vehicle in these black diamonds, it's already hypersensitive to bleomycin and the double-strand breaks because it can't do non-homologous end joining as a function, or classical non-homologous end joining as a function of the lack of Ku. And when we incubate our small molecule Ku inhibitor, in fact, we get only a modest increase in sensitivity, about 20% compared to a 5-6-fold in our wild type cells. So this tells us two really important points. One is that the cellular effects we're seeing are dependent on having Ku in the cell, which is nice because we develop these as Ku targeted small molecule inhibitors. And also that the inhibitors that we have are giving us a degree of sensitization that is not quite to what we see in a Ku null cell. So that says there's opportunity for increasing the potency or increasing cellular uptake, making better molecules that can block this even further to get to that Ku null phenotype. But we've got on-target activity. We have the ability to inhibit the autophosphorylation events, as we mentioned in cell-free extracts, as well as in the purified proteins, and sensitize cells to double-strand break inducing agents. So while MEFs are a nice model, one of the questions was, could we do this in something more relevant in the context of cancer therapy? So we took a H460 non-small cell lung carcinoma, so now KRAS mutant, all the problems associated with a standard cancer cell line model. We can demonstrate the same thing now in the case of 245, very efficient sensitization to bleomycin-induced cell death. And in this case, we sought to correlate the phosphorylation, or the autophosphorylation of the DNA PKCS 2056 cluster, and see if we were seeing that in the cells as well to correlate with the sensitivity. And so in this reaction, in this experiment, Pammy Mendoza, a postdoc in the lab, assessed the concurrent treatment with pre-incubation, because we had seen the effects of enhanced efficacy in experiments where we do pre-incubation with the agents. And she was able to measure the DNA PK-CS autophosphorylation as well as gamma H2AX as a measure of the DNA damage response. And so qualitatively, it looks like we're getting less green down here and less red compared to the controls. Quantitation of that in a high content imaging analysis proved the point pretty convincingly. Looking at the autophosphorylation in the absence of any bleomycin, we get a base level. When we put in bleomycin, we can increase that. Control experiments performed with just the inhibitor alone have no effect on autophosphorylation from this baseline that we see. And when we do concurrent or a short pre-incubation, we actually get a pretty significant, statistically significant decrease in that autophosphorylation event, effects that are mimicked through the measure of gamma H2AX as well. So we're able to put this into these cells and demonstrate the effect. We've now done it with all 4 compounds, the 68, 132, 322, and 245. Some of it we've published recently in NAR and others are unpublished, but we get a very consistent phenomenon. So while bleomycin is a nice radiomimetic agent, obviously in the context of cancer therapy, about half the patients with solid tumors are going to see radiation in some point in their treatment regimen. And so we did the experiment measuring radiation sensitivity, and in this experiment show the 322, 245 molecules run in this type of analysis, essentially plate cells, treat them with ionizing radiation, trypsinize reseed count colonies, and either pre-incubate according to this schedule, 2 hours prior to the IR or with vehicle alone. And we get a statistically significant increase in radio sensitivity, a factor that's exacerbated even if we go out to 10 gray. But we wanted to have cells to look at when we do the analysis for immunofluorescence, measuring autophosphorylation as well as H2AX. We get the very uniform kind of punctate staining that you see with typical ionizing radiation and DNA PKCS autophosphorylation, as well as the H2AX. The high content imaging of these analyses demonstrated, if we're looking at DNA PKCS autophosphorylation, now the total integrated intensity was too low to test above background, so we ended up counting PKCS foci per nucleus. Untreated, there you get none. 245 has no effect. Ionizing radiation gives us about 50 per nucleus on average. And the pre-incubation with 245 drops that down to about 25, again, statistically significant. The NU7441 drops that completely. And so this was incubation with the positive control catalytic subunit inhibitor at a concentration of 5 micromolar. And so using
John,
less
your time is up.
Okay, I'm almost done here.
Okay.
We are demonstrate that we can block that phosphorylation activity and ultimately obtain the radiosensitization through this inhibition of Ku binding DNA PKCS autophosphorylation to increase cellular death. What I'll do is pass that and just go to our conclusions. We've developed these small molecule inhibitors. They can inhibit DNA-PK activity through this novel mechanism, which is blocking the Ku-DNA interaction. And we can abrogate autophosphorylation, and this could potentially be useful as a therapeutic strategy in the context of ionizing radiation, as well as the potential to help elucidate some of the internal mechanisms of homologous recombination, crossover with NHEJ, and potential telomere effects. People that did the work, Pammy Mendoza is a postdoc who's done a lot of the autophosphorylation. Pam Vandiver Carroza did all of the in vitro and in vivo analysis, and Katie Puelzak at Nerex Biosciences has done a whole bunch of work on the stuff I didn't get to show you. So with that, I'll stop and be happy to take questions if there